Hey students, so this is microbiology and chemotherapy, and the course code is CAM445, and this is lecture 8, and the topic is fermentation technology, primary and secondary metabolites. And I am Dr. Siddiq Ibrahim, Assistant Professor, Department of Chemistry, Kohart University of Science and Technology. And you can access the link of these lectures by logging into your KCMS account, and then you can browse to the Quest Media server portal, which is ms.cohort.edu.pk. Now, the topics to be discussed in this lecture include microbial metabolites, growth curve of microbes, primary metabolites, and secondary metabolites. Now, moving towards microbial metabolites. So, during the metabolism of microbial cells, a number of metabolites or compounds are produced, and many are secreted out of the cell. As we have discussed earlier, that by the process of fermentation, penicillin is being produced by Penicillium crassugenium, which is basically a fungi. So the penicillin is a secondary metabolite, and it is secreted out of the fungi cell. So it means that many metabolites which are secreted by the microbes, they are beneficial for human beings. Now, these metabolites or compounds, they can be easily extracted and are very useful for many animals from the point of view of treating infectious diseases. Now, the process of fermentation, which is carried out on the microbial cells on an industrial scale in order to get various metabolites. So the function is, the purpose is to use the fermentation technology by which the microorganisms are grown, and when the microorganisms are growing exponentially, and when they reach to the stationary phase, they then start to produce the secondary metabolites, and these secondary metabolites are then used for different purposes, either for treating infectious diseases or within the cosmetic industries, depending upon the nature of the compounds which are being produced by the microbes. Now, moving towards the growth curve of the microbes. So in this graph, which shows the bacteria population growth curve, you can see that on x-axis you have the time duration, and on y-axis you have the number of cells increasing. So this red line is giving you information that what is the mechanism by which the microbes are growing. So the first phase is the lag phase, so and it is up to this point, and in lag phase the microbes, they are preparing themselves so that they can start the growth. Then you have the exponential phase. So exponential phase is that phase of microbes in which the microbes are growing exponentially. That is, for example, if there are 2 microbes, so they are converted into 4, and when there are 4 microbes, they are converted into 16, and so on and so forth. So After a specific period of time, the exponential phase stops and the stationary phase of the microbes starts. Now, in stationary phase, the microbes are not growing exponentially, but in that phase, the secondary metabolites are being produced. And then you have the death or decline phase. So here you can see that with the passage of time, the cell decay starts and eventually the cells are dead. So this population growth curve is for the batch culture in a closed system of the fermentation along with the liquid medium which is provided as a nutrient for the microbes. So by seeing this growth curve, you can understand that there are basically four phases. The first one is the lag phase, the second one is the exponential phase, the third one is the stationary phase, and the last and the fourth phase is the death phase or the decline phase. Now, as we have discussed the growth curve for the microbes, now we are moving towards the growth curve for metabolites, which are also produced during the life cycle of the microbes. So this graph shows the relationship of production of primary and secondary metabolites with growth of the producing organism. So on x-axis you have the age of microbial culture, which is basically the time duration during which the microbes are growing along with the metabolites. And on y-axis you have the amount of growth and metabolites. So it means that either the microbial cells are growing or the primary metabolites are growing or the secondary metabolites are growing. So if we look at the first line, which is this one, so this line shows the growth of the microbial cells, which is pretty much similar to the previous slide in which we discussed that the microbial cells, they have a lag phase, then they have a exponential phase, and then they have a stationary phase, and then ultimately they have a death phase or declining phase. Now, in parallel to the growth phase and growth curve, you can see that there is a primary metabolite curve. So the primary metabolites, they are starting to be produced by the microbial cells after a certain period of time when the microbial cells have started growing. So along with the growth of the microbial cells, the primary metabolites are also growing and up to a maximum of the stationary phase. So when it reaches to the stationary phase, so then the primary metabolites, they are also in the stationary phase. And the important thing here which needs to be considered is the growth curve of the secondary metabolites. So the secondary metabolites, they start their growth after a certain period of time, and if you compare that at what stage the secondary metabolites, they start to grow, you can see that they start to grow when the growth curve of the microbial cells is in the stationary phase. So it means that the secondary metabolites, they only start their growth when the microbial cells have reached the stationary phase. Now, along with the growth of the secondary metabolites, they start to grow up to the maximum even when the cells, the microbial cells, they start to die out here. So you can see that the graph of the secondary metabolites is going upwards in this direction, while the graph of the microbial cells is going down. Now, discussing the primary metabolites. So, the primary metabolites, they are involved in the growth of the microbial cells, the development of microbial cells, as well as the reproduction of the microbial cells. So it means that the primary metabolites are very important for the survival of the microbial cells. So essential for the survival as well as the existence of the microorganism and for the reproduction of the microbial cells. Now these primary metabolites, they are formed at the same time as the new cells are forming by the process of exponential growth, which we have discussed in the previous growth curve slides. The production curve follows the growth curve. So it means that the production of the primary metabolites is happening at the same time as the growth curve of the microbial cells is taking place. Now these primary metabolites, they are also known as the central metabolites, as they maintain the normal physiological processes. Now the normal physiological processes include the DNA replication, the process of transcription, the process of translation. So if these primary metabolites are not present, then the survival and existence of the microbial cells is not possible, and therefore they are in the utmost need of the microbial cells. Now the primary metabolites, as we have discussed and studied, that they are very important. So the microbial cells, they maintain the optimum concentration of all the macromolecules which are responsible for the growth of the microbial cells. For example, the proteins, the DNA, RNA, and other macromolecules. Now, these primary metabolites are produced in adequate amount to sustain the cell growth. For example, the vitamins, the amino acids, the nucleosides, they are the primary metabolites, and they are needed for the sustained growth of the microbial cells. Now the examples of primary metabolites also include ethanol, citric acid, glutamic acid, which is obviously an amino acid, lactic acid, which is a kind of a carbohydrate precursor, acetic acid, acetone for the formation of ketone bodies, formic acid, butanol, propionic acid, dihydroxyacetone, which is again responsible for the formation of ketone bodies, and glycerol, which is also an example for the fat molecules and other molecules. So from this you can understand that the primary metabolites, they are very important for the sustenance and maintenance of the microbial cells. Now moving towards the primary metabolites and their importance in the industries. So there are certain primary metabolites which are important from the industrial point of view. For example, ethanol, acetone, lactic acid, and carbon dioxide. The common food supplements, like L-glutamate and L-lysine, which are basically amino acids. These are produced and purified by the mass production, large scale production of Corynebacterium glutamicum. So you can see that the amino acid like L-glutamate and L-lysine, they are produced by the specific bacteria which is called Corynebacterium glutamicum, and these are the primary metabolites in the life cycle of the microbial cells. Similarly, other important primary metabolites, for example, citric acid, it is commonly used in pharmaceutical as well as the cosmetic industries and is produced by Aspergillus niger. Now, one thing which is important to observe is that growth rate can slow down due to the limited supply of any other nutrient. So the metabolism, it does not stop, but the product formation is stopped. So what does it mean is that if you see the growth curve of the microbial cells, so at a certain point, due to the limited supply of the nutrients, the growth rate can slow down, which means that the cells, they are not growing at exponential rate. But the metabolism is not stopping within the microbial cell, as the secondary metabolites examples is in front of you, that they start to be produced when the stationary phase of the microbial cells is starting. So the metabolism does not stop, but the product formation of the primary metabolites, it stops due to the limited supply of the nutrients. Now moving towards the secondary metabolites. So the secondary metabolites, they are not produced until the microbe has largely completed its logarithmic growth phase and entered the stationary phase of the growth cycle. So this we have discussed in the growth curve of the microbial cells along with the growth curve of the metabolites, that when the microbial cells, they reach the stationary phase, so then the secondary metabolites, they are starting to be produced. Now the period of production is called idiophase, and the metabolites are known as idiolytes. So it means that the secondary metabolites are also known as the idiolytes, and the phase in which they are produced is known as the idiophase. Now in the idiophase, the cells, they do not divide, or the growth of the cells is not taking place, but the cells are metabolically active, which means that the metabolism is going on inside the pre-existing cells. Now the idiolytes, or the secondary metabolites, these are the organic compounds which are produced only after considerable number of cells and primary metabolites have accumulated. So it means that when the microbial cell growth reaches to the stationary phase, and there are maximum number of microbial cells along with the maximum number of primary metabolites present within the media, so then the idiolytes or secondary metabolites, they start to be produced. Now these secondary metabolites are produced under suboptimal concentrations of oxygen. So the suboptimal concentration means that the concentration which is less than the optimum concentrations. And also the deviations of pH. So as a result of accumulation of microbial cells along with the primary metabolites, there is some deviation of pH is taking place within the fermentation tank, or when the primary nutrient source is depleted. So the nutrients, the media which is provided as a source of food for the microbial cells to grow. So when the microbial cells they reach the stationary phase, so at this stage the primary nutrients are all being consumed out, and then the secondary metabolites they start to be produced. Now the secondary metabolites, though these are called ideolites, and these are the specific or characteristic feature of fungal, yeast, actinomycetes, and bacterial growth, but in some instances they are not produced by a few strains of E. coli. E. coli stands for Escherichia coli. So it means that there are certain microorganisms which are not producing secondary metabolites, but most of the fungal, yeast, actinomycetes, and bacterial species they produce the secondary metabolites. Now, in some strains, the secondary metabolites are produced by the conversion of primary metabolites. So it means that when the primary metabolites are being produced or have been produced, then the primary metabolites are being converted into secondary metabolites, but that is possible in some strains of microorganisms. Now, as we know that the secondary metabolites, these are not necessary for growth, development, and reproduction, as for the case of primary metabolites, which are important for all these three processes. But the production of the secondary metabolites is influenced by environmental factors like pH or suboptimal concentrations of oxygen. Now, the secondary metabolites are synthesized for a specific period by the cells which are no longer undergoing balanced growth, which means that the cells, the microbial cells, are not dividing uh, exponentially, which means that they have reached the stationary phase, and now they are going to die. But before dying, the microbial cells they are producing the secondary metabolites. Now, in case of secondary metabolites, they are very complex type of metabolites, and a single microbial type it can produce very different metabolites which can be used for different types of purposes, either in the cosmetic industry or in the pharmaceutical industry. Now, the production of the secondary metabolites is regulated by complex biochemical pathways, and some of the microbial strains can produce a variety of ideolites or secondary metabolites. For instance, a strain of Streptomyces can produce a variety of 35 types of anthracyclines. Now, the typical examples of secondary metabolites which are produced by the microbial cells, they include antibiotics, toxins, and pigments, to name few. Now, for example, bacitracin, which is a secondary metabolite, uh, is actually a peptide antibiotic, and which is being obtained from Bacillus subtilis and is commonly used as a topical drug in the form of a cream or ointment. And bacitracin is synthesized in the nature through the non-ribosomal peptidase synthetase enzymes, which are abbreviated as NRPSS. Now, this bacitracin it has a wide spectrum against gram-positive as well as gram-negative bacteria. So it means that bacitracin in the form of a drug is effective against gram-positive infections as well as the gram-negative infections. Now, in case of secondary metabolites and their importance, the few examples of secondary metabolites in the form of antibiotics and their microbes which are producing them is given below. So, for example, the antibiotic amphotericin B is produced by Streptomyces nodosus in the form of a secondary metabolite. Another example is chloramphenicol, which is a very famous antibiotic and is produced by the microbe known as Streptomyces venezuelae. Another example is erythromycin, which is produced by the microbe known as Streptomyces etheris. Now, gentamicin is produced by Micromonospora purpurea. And another example is gramicidine, which is produced as a secondary metabolite by Bacillus brevis. Penicillin, which is a very common example, and we have discussed its production by the process of fermentation, is produced by Penicillium chrysogenum. The streptomycin, which is also a very famous antibiotic, is produced as a secondary metabolite by Streptomyces griseus. And tetracycline, which is also a very effective antibiotic, is produced by Streptomyces aureohascens. So these are the references, and for further reading and explanation you can consult these books.